All right, so we're going to start up now with our second section on viruses. And for the first little bit of this section, we're going to take a look at, just kind of from a very general perspective, how exactly viruses do what they do and how they actually cause harm to the organisms that they infect. So, really, when you think about a virus, all you think of is basically a set of instructions on how to make more viruses. That's really all it is. The virus gets into a host cell and takes over that host cell, causing it to make more viruses, which of course then get out and attack more host cells and cause them to make even more viruses and so forth. So that's basically the idea, because like we talked about back in our first section, outside of a host cell, a virus is just a couple of molecules, really. It's some DNA, it's some protein, at least at a bare minimum, and that's really all there is to it. But once it gets into a cell, then it's a very different story because these viruses can, like we said, trick these host cells to start making more viruses, which can then, again, in turn infect more cells and then cause them to make even more viruses. So, because viruses are acellular, meaning that they don't have a membrane, they don't have cytoplasm, they don't have any ribosomes, they can't make their own proteins, they can't make their own DNA for that matter. They need a host cell to do that for them, which is the whole reason why they infect host cells to begin with. So, viruses contain different sets of genes that allow them to infect a host cell at different stages along the process. So, early genes, middle genes, late genes. These are essentially what allow viruses to get into a host cell, to take over the host cell, to destroy the host cell, and therefore release more viruses, those kinds of things. So we kind of think about their genome as being broken up into these early, middle, and late genes, which allow them to perform their job, which is to eventually allow for the assembly and eventual release of even more viruses. Now, not all viruses cause disease. Many viruses can go into what's called a latent period, which is a period where they're infecting a host, they're in host cells, but there's no noticeable symptom present. There's no sign of anything being amiss. There's no damage being caused. There's no fever being induced in the host cell. It's just a really business as usual sort of a thing for host cells that have these latent viruses. Viruses typically are very, very specific to individual host species. They typically do not cross species lines, although they can, at times, pick up a mutation which can cause them to switch hosts. And that's really what we get into when we start talking about these really big, scary, what we call emerging viruses, is viruses that are normally present in other species that actually jump then into our own species and therefore can cause some very, very dramatic damage as a result. Now, for that to work, they have to be fairly closely related, right? So we're not going to see like a fish virus or a snake virus get into humans. Mammalian viruses, like from bats, for example, or sometimes bird viruses, like we saw with the so-called swine flu back a few years ago. They're a little more closely related to humans, to us. So it's a little bit easier for them to pick up that mutation, which allows them to jump into that human host. But even still, that doesn't happen very frequently at all. It's a very uncommon sort of a situation, actually. The more the more we have animals, especially animals that are from the wild and carrying perhaps viruses that we don't know about, the more we have those kinds of things in contact with humans, the more opportunity there is for these things to happen. But typically, it's not a very common situation for these species to pick up viruses from other species. Even within a host species, viruses are very specific to certain kinds of tissues. They exhibit what's called tissue tropism, which is where a virus will only attack a certain kind of tissue in the body. So, for example, HIV only attacks certain T cells, and polio, for example, only attacks neurons in a certain way. So, even within a host species, the viruses that are there are very, very specific to certain parts of that host that they can then infect and cause damage to. Polio, like we talked about, is a neurotrophic virus, and polio is actually a really good example to talk about for a couple of reasons because back before the 1950s or 1960s, polio was a very, very real concern for most people in this country and in others because lots of people got polio, and polio could cause some very severe neurological damage. It could cause paralysis. It could cause all kinds of horrible symptoms. And in some cases, it could even cause death. So, if you were to ask your grandparents, great grandparents, and so forth before that about polio, they would have been they would have known about it and they would have been very scared of it because everybody knew somebody who had polio. Maybe they had polio themselves, and it was a really, really nasty disease. Now, the good news about polio is that there's a very, very effective vaccine against it, and in most parts of the world, that vaccine has been deployed very, very well. And so polio doesn't really exist so much anymore in the human population because the vaccination has allowed it to sort of dwindle away to almost nothing. There's a couple of countries that still have it here and there for reasons that are more social than biological or medical, but for the most part, we don't really see polio showing up so much anymore in human patients. Now, another feature of viruses that we sometimes talk about is symmetry. Right? So, for example, viruses like that tobacco mosaic virus that we talked about back in our first section of the lecture is a helical virus because it's got sort of a spiral sort of symmetry to it. Binal symmetry viruses are those like this bacteriophage here, where you can draw a line of symmetry down through the middle and get equal left and right sides to it. A lot of bacterial viruses or bacteriophages have that sort of symmetry where there's just basically one line of symmetry there. Polymorphic symmetry is a situation where there is no distinct symmetry. The viruses are just sort of amorphic. There really isn't a consistent, predictable shape to their capsid or anything like that. They're just sort of without a typical form. Icosahedral symmetry is a situation we see a lot with animal viruses where a virus will have multiple sides to it, almost like one of those really fancy dice for like Dungeons and Dragons, those kinds of things, where it's got like 22 sides to it. That would be an icosahedrally symmetrical virus as well. So we see, variation here in symmetry. Influenza is a pretty common one that we talk about a lot, right? So this is a helical capsid within an envelope. So this one's a little more complicated because it does have that envelope structure we talked about, sort of the way for it to hide out from the host immune system. And then within that, there's a helical or spiral-shaped capsid, which is this part. And then within that, there's the RNA, which we see here. So that one's a little more, complex than some of the other viruses that we sometimes look at. Viruses, very small, right? So we take a look just for scale here at the size of a eukaryotic yeast cell. So yeast cells are fungi. They're not very big. They're only 7 millionths of a meter long. But compared to viruses, the yeast cell is absolutely massive. I mean, even bacteria compared to viruses are huge. So pox virus, herpes simplex, we're down in the range of nanometers here. So billionths of meters or less in some cases. So we get down to, the yellow fever virus, which is 22 nanometers. That's just barely bigger than a hemoglobin protein inside of a blood cell. So, some of these viruses are absolutely tiny, and for that reason, very difficult to sometimes avoid, as we see with the COVID-19 situation that we're experiencing right now. As far as the number of genes in a viral genome, typically it's not very many. You might be looking at single-digit genes, maybe a couple of dozen genes at most. RNA viruses, in particular, tend to be single-stranded, although there are double-stranded RNA viruses, and there are both single and double-stranded DNA viruses as well. RNA viruses tend to be single-stranded, and because they're single-stranded, that really lends them to a relatively high rate of mutation, right? So when we think about DNA replication, we talked about this before several chapters ago, where you take one parent strand and you use that as a template to build a daughter strand, and that doesn't really leave a whole lot of room for error. Because if the parent strand has a C on it, we know the daughter strand has to have a G on it, and so forth. If it's just a single strand of RNA, however, then there is no template strand to use in the same way that we have when we copy DNA. So RNA viruses are much, much more prone to mutations or mistakes as they're being copied than are things like DNA viruses. So that gives them a relatively high rate of mutation and also a relatively fast evolutionary rate as well. That makes it very difficult for our immune system to fight them if they're always changing. It makes it very difficult for vaccines or antiviral drugs to fight them off. Again, if the virus is always changing, it's like hitting a moving target. Right? You could spend anywhere between 3 to 5 years making a vaccine, testing it, marketing it, getting it cleared by FDA and so forth. And by the time you actually make this vaccine that's cost you a couple $1 billion to develop as a pharmaceutical company, there's a really good chance your virus may have changed and that vaccine may not do anything against it. So it's a really difficult thing to fight off these viruses that are, on one hand, it's not really to their benefit to make a lot of mistakes because most of those mistakes probably don't work very well. But for those few mistakes that do work, that gives them a very, very big leg up on survival when it comes to dealing with host immune systems and also our attempts to cure them medically. So when we're looking at viral classification, the first thing that typically virologists would look at would be whether a virus has either DNA or RNA as its genetic material. From there, they would then try to figure out if it's single-stranded or double-stranded. Because once we know that, if you sort of imagine a situation where somebody brings in an unknown virus, and it's your job to categorize it or classify it as what kind of virus it is and how it might behave, and what to expect from it, what we predict might happen to a patient that has it, and so forth. The best approach is to take the virus and compare it to other viruses that are similar and that we already know something about. So by comparing it and finding out that, oh, it's a double-stranded DNA virus or a single-stranded RNA virus, then we can compare some known double-stranded DNA or single-stranded RNA viruses and see how they behave and see what kinds of effects they have on the immune system and see how fast they might evolve. It's like, you don't want to start from scratch every time a new virus is discovered. You want to take what we already know about other viruses and use that when we're trying to figure out how this new virus might behave. We could also take a look at the structure of the capsids, compare that to known viruses as well. We could look at whether or not it has an envelope. We could look at the symmetry of it. All of these things are going to tell us a little bit about what this virus is and how it might behave. Right? So again, rather than starting from scratch and reinventing the wheel every time a new virus is discovered, we take what we already know about pre-existing viruses and use that to predict how a new virus might behave. Now, the next thing we're going to look at is a typical viral replication cycle. All right, so the replication cycle that we're going to look at occurs in a bacteriophage, which we've used that term a couple of times already, but a bacteriophage is a virus that infects a bacterium and does basically the same thing it would do to a human cell. It takes it over, it tricks it into making more viruses, and eventually causes damage or death to that host cell as the new viruses are leaving it. So the very first step in this viral replication cycle is for the virus to gain entry into its host cell. And so the first thing that needs to happen is the virus needs to attach itself to the outside of a host cell, and from there, gain entry to it. Now, again, these viruses are very, very specific to certain kinds of cells and certain kinds of tissues. So for a cell to be acceptable to a virus, it has to have the right kind of surface receptors on it. The right kinds of proteins on the outside of the host cell will then let a virus know that that host cell is suitable, it's appropriate, and therefore it should attach and try to move on to the next step. If it doesn't have the appropriate receptors, then the virus is just going to continue being inert, move on through the environment until it comes across, hopefully for the virus, the right kind of cell with the right kind of receptors. All right, so we're going to assume that the cell's found the right kind of cell with the right kind of receptors. So the next step that's going to happen is the virus is then going to inject its genetic material into that host cell. It's like with almost like a hypodermic needle situation where the DNA is injected into the host cell, the protein coat stays on the outside and eventually just sort of sloughs off and goes off into the environment. If we remember Hershey and Chase experiment back from chapter 14, where they used the radioactively labeled viruses with the radioactive phosphorus in the DNA and the radioactive sulfur in the protein, and they looked to see what happened to the bacterial cells where they picked up radioactive phosphorus or sulfur. It was always radioactive phosphorus because the virus is always injecting its DNA or RNA into the host cell. And DNA has phosphorus in it, whereas proteins don't. Proteins have sulfur in them, whereas DNA doesn't. So that was kind of the recap of what we saw with Hershey and Chase, but that really kind of goes back and reinforces what we're dealing with right now. So once that DNA or RNA has been injected into the host cell, basically what's going to happen is everything that host cell was doing up to this point is now going to be put on hold. And it's not really going to have much ability to do much else except make new viruses. Okay, so essentially what's happening here is that the viruses are hijacking the host cell. They are causing the host cell to put all of its own activities on hold, and that host cell is going to start making some new viruses. Okay, so it's now in a viral producing mode. It's been taken over. It's like a factory that's been hijacked, and it's being used to make more of these invading particles. So the next step after that, once the viral particles have been built, is for them to be assembled. So the viral nucleic acid is going to be packaged up with viral protein, and that's the assembly phase of the reproductive cycle. The very last step is going to be release, and that is the release of mature viral particles into the environment where, again, hopefully for the virus, it's able to find new host cells and then perpetuate this cycle even further. Of course, most of those viruses may never find a host cell. They might just sort of remain in the environment until they're inactivated by some sort of inhospitable feature of the environment, but some of the viruses might find a new host cell and then restart the cycle from there. The eclipse period is the time between the attachment phase and the formation of the new viral particle. So basically steps 1 through 3 is the eclipse period. During that time, if the host cell is destroyed, not a whole lot happens as far as the virus is concerned because it hasn't been assembled yet. So it's just sort of a cutting off the cycle halfway through sort of a step. Now, something else to think about here is, okay, well, that's all well and good, but why does this make us sick? Why does this cause us to have symptoms? Why, why does this cause harm to the body to have these viruses present? And the answer to that is, if a host cell, if our cells are no longer able to do what they're supposed to do, right? If they're no longer able to carry out functions that the liver cells would normally function or neurons would normally do when they're functioning, then those cells' failure to function is going to lead to the production of symptoms in the host. So when the viruses are taking over the host cells, the host cells are unable to do their job, and that's where we start seeing symptoms showing up in the host. We can also see some of the effects of the body's response to the infection by producing a fever or those kinds of things. But really, the damage that's done to a host is done because the host cells are no longer performing the functions they're supposed to function, because they're devoting their lives 100% now to the formation of new viral particles. So that is essentially a description of what we call the lytic cycle in virology. The lytic cycle involves damage to the host cell. It involves the assembly and then release of viral particles, which destroys the host cell in the process, releasing new viruses. So that's a pretty typical sort of a cycle. We also say these viruses are virulent because that means they're able to produce disease. There's also what's called a lysogenic cycle, which is a little more advanced. And this is where viruses will actually take their DNA once they get into a host cell, and they will make their DNA or RNA part of the host cell's DNA. All right, so they actually take their genetic material and they incorporate it into the host cell's genetic material. And then they're just kind of along for the ride at that point, right? So if you think about the whole point of natural selection and the whole point of being adapted, and the whole point of being fit is to make sure your genes are passed on. Well, for a virus that puts its DNA into a host cell's DNA, every time that host cell divides, it's also going to copy the virus's DNA and pass that on too. So what better way would there to be fit than for a virus to just take its DNA, put it into a host cell's DNA, and let the host cell do all the work throughout the generations? And so these viruses can hang out in host cells for dozens or hundreds of generations of host cells. They may never come back out again as far as being, separate pre-existing viruses, but they can sometimes switch back to being lytic, as we'll see. So these viruses actually, when they're incorporated into the host cell's DNA, we call them prophages. And again, during that time, they're not really causing any symptoms. It doesn't really cause any harm to the host. It's an almost guaranteed way for the virus to make sure its DNA or RNA gets passed on to the next generation of viruses, because it's going along with the host DNA in the process. All right, so that's kind of like a quick overview of lytic versus lysogenic viruses. These pictures here are electron micrographs of bacteriophage, so bacterial viruses that infect bacteria, in other words, that are attaching to the outside of this bacteria. This big bacteria is about to have a really bad day because, first off, it's already filled up with a bunch of viruses that have been made, and it's still being attacked by other viruses on the outside. So this bacteria in this picture is not long for this world by the time that picture was taken. But that's, a pretty typical situation for how bacteriophages might behave. All right, so I think what we're going to do here is we're going to cut off this section, and we'll start up in just a few minutes with our third section on viruses.